So you just instigate that stroke to come along if somebody has a low blood platelet. People like that have to take a blood transfusion first and then go to the Chondriana treatment, so that in order to avoid to getting the stroke. And that's why the clinic's afraid and they don't want to use Chondriana when people are in such a critical stage. They will try to nurse back the organs and restore the system so that people can accept the Chondriana. Now, as you said, if you got chemotherapy a year before, I mean somebody, apparently, you know, there's a lot of damage has been turned around and repaired.
Yes.
In that case, those are the one who comes out from, according to the statistic, those are the one who comes out and the Chondriana is successful after chemotherapy treatment. But most of the one cannot take the Chondriana because of the conditions they are because of the treatment.
And you're not differentiating between different types of chemotherapy, 5FU compared to a different type of chemical?
Of course, they're all different. They all have work different, and they all you know, producing different kinds of damage depending on what it is. 5U4 is also very bad.
Okay. And has any work been done with the Chondriana in primary biliary sclerosis?
Primary what?
Biliary sclerosis?
I'm not familiar with it.
Okay, so how would we access information about that?
Well, you will have to talk to the clinics or the doctors. So the only area I know when they're sending me the specific reports and specific cases, because you're collecting all the clinical data.
Okay.
And you know, where they try to go after the main untreatable diseases, which is necessary to prove it for approval.
All right.
So they don't go after every disease. But if some comes along, you know, they will not turn the patients down. But I don't have anything on that particular one in my file.
Thank you.
You're welcome.
My doctor has said that he would be willing to inject me with this Chondriana if he has information. Is that possible for him to do it, a doctor in the Lower Mainland, or do I have to go to a clinic that's already established and using it?
No, your doctor can do it. Your doctor should get the protocol, which is supplied by the suppliers. And there's also a clinical data book floating out there somewhere, and that available, so he can go through and see what other doctors have done in different parts of the world. It's describing the treatment. I would be very happy to talk to him too, based on my experience. And based on the protocol, he could treat you. And any doctor knows the basic function of the body and the organs, so they know what to do. Okay.
And we would access you then through your state's number, or how do we?
No, you access directly to the number I was just giving to Mexico, to Dr. Villapando or Dr. Devgan in Toronto.
So that's how I access you, through those numbers. Thank you.
Yes, correct. Thank you.
I know a lot of people are trying to get this information of accessibility and so on, and I respect that. And I don't want to take too much time talking about the basics, but when you started, or I've seen your video, which show what happens in the life of the cells at a very, very high magnification. And my burning question is, how does your work overlap or add to the work of such other researchers that work with dark field microscopes, notably Dr. Gaston Naissons here in Canada, who has isolated what he called somatids, and which are presented as being, you know, very simple, and like in a simpler form, at least in a popular form, described in a book by Christopher Bird, a popularized way of presenting.
Familiar with it, yes.
Yes, and a lot of I bet that a lot of people here in this audience have become, to a small degree, educated in, you know, through that book. So, are you if you're familiar with Gaston Naisson's discovery, then how does your discovery overlap?
I also, that doesn't overlap at all. I also replicated his somatids, and remember, somatids, a submicron organism. A Chondriana is 100 micron. A Chondriana as ancestors or precursors of human beings. Some of it has not been made by human genetic material. It may be made by human genetic material because it lives in the body, but not like the way Chondriana is made, and they're not related at all. If you go into the bloodstream, or even if you go inside the cell, there are as many life forms and as many colloidal life forms than suns and stars in the whole universe. There's so many of them. Now, his contribution to the medical science of biochemistry, that he discovered that anybody who has cancer, the somatids in their blood go through the bacterial cycle, and I verified that for him because I reran his process. I replicated his process with the somatids, and the only difference is that what I did, when the somatids get into the ASCII cycle and they produce spores, I took some of these spores and put it together with the stem cells. And when these stem cells gets into, these spores gets into a stem cells, they disrupt the genetic process. It blows up like a balloon, and you will see it on my yellow book, and out comes 6 malignant cancer cells. Wow. So the spores of the somatids actually can cause cancer. This is why what he discovered, that people who have cancer, they have somatids in the bacterial cycle, and he deserves all the credit for that. But that gives you another angle to look at, because the cancer epidemic is on top of us. And in this decade, one out of every third person in this room is going to have cancer in this decade. Now, there must be many reasons for it. Cancer can be caused by many ways. Radiation disrupting the DNA chain, so the DNA replicates itself faster, and the cell will reproduce much faster, proliferate much faster. So the cancer cells really premature stem cells, and you can cause the same breakage of the genetic material or damage to the genetic material by chemicals. But spores, if the spores gets into, which I proved, that spores gets into stem cells, can differentiate into cancer cells. And these spores not necessarily coming from the ASCII in the somatid cycle, but rather penicillin, antibiotics, and all the drugs that manufacture with fungal or yeast processes. And they all contaminated with spores. So that the sources of the cancer can come into from many different directions, because the environmental condition we live and the toxic food, water, and chemicals and medicine we are producing.
Further, I'm wondering, there's something that Gaston Naissance is doing right, it appears. He's injecting, I believe, some camphor tree derivative right into the lymph nodes of his patients, and claims a very high recovery rate in 750 out of 1,000 cancer cases. Is he inadvertently affecting the presence of life crystals or of
no, I don't think so.
What's happening there then?
Well, I don't know how his system works. He developed his system. And all I can tell you that I gave a lecture in London, the Royal Academy of Medicine in 1968. And at the same time, there was a Dillington Tea Company there promoting the camphor to cure cancer. So a lot of work has been done in England already then about the camphor. Now, how his system is working, he is the one to know, because that's his product, but I'm not familiar how his somatids working.